Last time எதை வச்சு எடுத்தேன்? Hi students, good morning. I hope you are all good in this morning. So before entering into the class, your exam is going on so soon. So on May it will start, right? So I think you are all prepared for your exams. Be prepared, keep on studying and give some small gap and take rest and study well, students. So in this morning I am just welcoming you all to our Future Doctor Academy. So let's studying the cell, the unit of life. So far we have finished the cell theory, what is cell and cell theory and cell modification and prokaryotic cells. So till that we have studied, and we just go through from yesterday's subject. Okay. So what is prokaryotic cells? So what are all the prokaryotic cells? Very good. Cyanobacteria, blue-green algae, bacterium. So PPLO, these are all the prokaryotic cells we have studied. So and then what are all the four shapes of prokaryotic cells? Okay. Very good. So coccus, bacillus, vibrio, and last spirillum. Okay. Then what will be the prokaryotes? Prokaryotes with cell wall is called what? Prokaryotic without cell wall is what? Prokaryotes with cell wall and prokaryotes without cell wall. So we didn't see in the last class this question. So it will be asking for a NEET examination. So prokaryotes with cell wall is bacteria. So bacteria is having a cell wall, right? And prokaryotes without cell wall is mycoplasma. So these will be the answers. Prokaryotic with cell wall is bacteria, and prokaryotes without cell wall is mycoplasma. And what will be the bacterial structure? What are all the two DNAs? Small circular DNA and genomic DNA. Plasmids are present in the prokaryotes, whereas plasmids are not present in the eukaryotic cells. Okay. Then what is the name of infolding structures? Infolding of cell membrane is what? Yes, we know that. So that is mesosomes. Okay. Okay. And what will be the three layers of bacteria? Bacteria we have studied. Three layers. Like this we have studied. Outermost layer. What will, tell. Okay, very good. So outermost layer is glycocalyx and followed by, innermost cell wall and followed by, what is this? Yes, very good, plasma membrane. Okay, plasma membrane. So glycocalyx, cell wall and plasma membrane. Glycocalyx is having a two layers. What are the two layers? Very good. Capsules and thick and rough layer is called capsules, and first one slime layer, okay, and that is a loose sheath. loose sheath layer is called slime layer, and thick and rough is called capsules. And what are all the extensions of mesosomes, that is cell membrane, plasma membrane? There are three types of extensions we have studied. What are all they? Very good. Tubules, vesicles, and then lamellae. So these are all the three. And what will be, it is helpful for three other functions. What are the three other functions? Cell wall formation, second one, cell wall formation, then DNA replication. In which phase DNA will replicate? In which phase? I have told, no? In cell division we are going to study the DNA replication. So DNA replication will be start at S phase. And then third one is distribution of daughter cells. Distribution of daughter cells. This is the three parts. And what will be the, if is the bacteria is motile or non-motile? Maybe motile or non-motile. Maybe motile or non-motile. So, and then the three parts of filament. Bacterial flagellum is composed of three parts. What are they? Filament, hook, basal body. Very good. So, filament, hook and basal body. And pillae means what? Pillae is, pillae and fimbriae. Okay. Very good. So, fimbriae is sprouting out, and pillae is formed of protein, synthesis protein. Okay. Now, ribosomes and inclusion bodies. What is ribosomes? We have already seen. What is ribosomes? Non-membrane bound organelle. This is okay. Very good. But present in both prokaryotes as well as in the eukaryotes. Okay. Here we are going to see the prokaryotes as well as the eukaryotes, because ribosomes is present in both prokaryotes and eukaryotes. So it is having some sedimentation. So in this sedimentation, it is called 80S or 70S. So what stands for 80S and 70S? What is this? S stands for what? Swedberg unit. So 80S and 70S stands for Swedberg units, because Swedberg was invented this one. So what is 80 and 70? Because here ribosomes will be this. So see, it is a chain. Ribosomes will be like this. So one will be shorter and one will be the bigger. So here it is having an 50 plus 30 combinations. So 30S and 50S. So altogether 80S. So where prokaryotes is having 80S or 70S, we are going to study. So this is the sedimentation rate. So sedimentation, Swedberg unit is right, but it is the sedimentation. While put the unit for the centrifuge. Centrifuge is one machine like substance. So it will be centrifuge. So in this centrifuge, the sedimentation rate timing will be as 80S or 70S. Okay, can you understand? Okay, let's study about the ribosomes. So in prokaryotes, so first we are going to see about the prokaryotes. In prokaryotes, ribosomes are associated with the plasma membrane. So in plasma membrane only, cell is having the plasma membrane, and in plasma membrane which is associated, this ribosomes is associated in prokaryotic cells. So next point, they are about 15 nanometer by 20 nanometer in size, and they are made of two subunits. So here it is having an 15 nanometer, this prokaryotic cells. So having 15 nanometer by 20 nanometer. So it is having some subunits, that is an 50 and 30S. So subunits is, this is 30S and this is 50S. Which when present together form, which was present together. So 50 plus 30 is 80. But in prokaryotic cells, only 70 subunits will be, Swedberg units will be present because the sedimentation time, that is an sedimentation time. So, it is having 70 S. And then prokaryotic ribosomes. Ribosomes are the site of protein synthesis. So, we all know that ribosomes has an synthesis of protein because it is having a ribose sugar. So, here in endoplasmic reticulum, rough endoplasmic reticulum, it is having some ribosomes, proteins. So, this is the proteins. So, ribosomes, in the sense it synthesize proteins. Several ribosomes are attached to the single mRNA. Several ribosomes are attached in the single mRNA and form a chain called polyribosomes or polysomes. So, here first point is several ribosomes are attached in a single mRNA. See, this is single mRNA as a polypeptide chain. Several ribosomes. This is ribosomes. So, several ribosomes are present in single polypeptide chain like mRNA. This is mRNA. This is called polyribosomes because too many ribosomes are here, not only one ribosomes. So, too many ribosomes is called polyribosomes or polysomes. The ribosomes of the polysome translate the mRNA into protein. This polysome is translation. Translation takes place here in the sense. Translation takes place. This mRNA synthesize what? Synthesize protein. This mRNA synthesizes protein. So, it is an in prokaryotes it is 70 subunits, Swedberg units. Okay. Then inclusion bodies. What is inclusion bodies? Inclusion bodies means what? Inclusion bodies. So, inclusion bodies means it is also an some organelles. So, it will reserve or it will store some parts. What are they? We show. So, reserve material in prokaryotic cells are stored in the cytoplasm. So, reserve material means what? Reserve material. Reserve material. Reserved means what? Store. It is storing some materials. What are all the materials it is storing? So, we are going to see. In prokaryotic cell we are seeing. So, inclusion bodies. These are not bound by any membrane system. So, it is not in this inclusion bodies is not bound by any membranes because ribosomes is lack of non-membrane bound organelles. And what is membrane bound? Eukaryotic is membrane bound organelles, whereas this is lack of membrane bound also. It is present both in prokaryotes and eukaryotes. But in the inclusion body, it is not an membrane, not any membrane system because it is lying free in the cytoplasm. If it is this is on cytoplasm, it will be free. Like freely this inclusion bodies will present. For example, phosphate and then granules, cyanophene granules and glycogen granules. So, what are all the inclusion bodies? So, for example, in your NCERT, it has given that phosphate granules, cyanophene granules and glycogen granules. These are all the granules present in the cytoplasm. So, and then gas vacuoles are found in blue, green and purple and green photosynthetic bacteria. So, what is gas vacuoles? So, vacuoles means bag-like structures. So, it is fluid matrix called cytoplasm, we have already seen. So, in this gas vacuoles means air will present. So, in this air present gas vacuoles we can see some organisms. So, what are all they means? Blue green and purple green photosynthetic bacteria. So, this is cyanobacteria. So, in inclusion body also it is present in cyanobacteria, blue-green algae. And then we are going to move on to the eukaryotic cells. So what is eukaryotes? Eukaryotes. Eukaryotes. Eu means what? Karyotes means what? Eu means true. And then karyotes means nucleus. Very good. So eu means true, karyotes means nucleus. Whereas in this eukaryote means true nucleus. Nucleus will be present in the eukaryotic cell, whereas it is absent in the prokaryotic cells. So in nucleus, what are all the parts will be present? Tell me. Okay. Then. So nuclear membrane, nucleolus, nucleoplasm. Nucleoplasm is also called as what? Chromatin reticulum. So these will be the these are all the things we are going to study in eukaryotic cells. So eu means true and karyotes means nucleus. So let's start the eukaryotes. The eukaryotes include all the protists, plants, animals and fungi. So includes all protists means what? Eukaryotes, the 5 families. What are all the 5 families in animal kingdom? So in this, which one is accepted? Monera, because Monera is having bacteria. Monera is having bacteria. So bacteria is an prokaryote. So without Monera, rest of the 4 kingdom will be in eukaryotic cells. Okay, that is protists, plants, animals and fungi. These 4 are eukaryotic except Monera, because that is an bacteria, it is an prokaryotes. Okay. So in eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane bound organelles. So we have already known that eukaryotic have an membrane bound organelles, and it is having a separate compartmentalization means separate. So separate compartment, separate compartment will be seen in the eukaryotic cells. And then eukaryotic cell possess an organized nucleus with a nuclear envelope. So what is nuclear envelope? Eukaryotic cell is covered by an cell complex, or that is nuclear envelope, that is nuclear membrane. Nuclear membrane is the nuclear envelope here. And in addition, eukaryotic cell have a variety of complex locomotory and cytoskeletal structures. So in eukaryotic have some varieties. What are all they? Complex locomotory. Complex locomotory means what? Absence of moving. They can able to move locomotory substance. They can able to move like a flagella. Flagella will be motile, right? So yes, it is because it is in complex locomotory, it gives some movements to the cell. So that is an complex locomotory, because in eukaryotics some varieties. The first variety is complex locomotory, and second one is cytoskeleton. What is cytoskeleton? Cytoskeletal. It gives a shape, structure. Cytoskeletal. It gives a shape and structure to the cell. And then their genetic material is organized into chromosomes. This locomotory substance, cytoskeleton, because in chromosomes it will be chromosomes will be present in the genetic material, that is DNA and RNA is present inside the chromosomes, nucleus and all. So it is a genetic material organized by chromosomes. This is the eukaryotes. So all eukaryotic cells are not identical. Why all eukaryotic cells are not identical? Because plant, animals, these will be separate, different. So it is not identical. It is not a twins, like identical twins. So plant is some other shape, animals is some other shape, whereas ourselves, human beings also, comparing to plants, human beings is separate, animals, yeah. So plants and animals is having and separate shapes. So eukaryotes have and not identical, different shapes will be present in the eukaryotic cell. That is, plant and animal cells are different in the former possess cell wall, plastids and large central vacuoles which are absent in animal cells. So animal cells will be absent in plastids. So prokaryotic cell, plastids will be present. In animal cells, plastid will be absent. Both are having ribosomes. Nucleus will be present. Nuclear will be absent, whereas eukaryotic nuclear will be present. And then one more. That is a lack of membrane bound organelle, whereas eukaryotic is membrane bound organelle. So these are all the differentiation. We will see after finishing this, we will see the differentiation. And then cell wall, plastids and large central vacuoles. Large central vacuoles means what? So large central vacuole means it is an sac-like cell organelles. In this cell organelle, some fluids, materials will be stored, that is central like vacuoles, that is large central vacuoles. So at that point we will be asking for your NEET examination, which are absent in almost all animal cells. So on the other hand, animal cells have centrioles which are absent in almost plant cells. So animal cell which is present? Centrioles will be present. Centrioles will be present. Where this centrioles will be absent in the plant cell. Then, so in your picture you are seeing diagram figure 8.3, that is one A is plant cell and B is animal cell. So in this vacuoles are present in plant cell, whereas you can't able to see in animal cells. So absent in animal cells, and in animals you can see centrioles, but in plant cell it was absent. And then chloroplast giving color to the plant, right? So it is present in the plant cell and it is absent in the animal cells. So these are all the differentiation. So you all people is going to tell the differentiation of eukaryotes and prokaryotes now. So first point, absence of nucleus. Very good. Next, here presence of nucleus. Then second, ribosomes will be present. Ribosomes, here I am writing this symbol. So ribosomes present, here also ribosomes present. Then this is an non-membrane bound organelle. This is membrane bound. Next. Plasmids present here, plasmids absent here, okay. Then next, vacuoles, here also vacuoles absent, right. Next. Next. Cell wall. Cell wall. Here absent. So plant cell wall present. Here absent of cell wall. Centrioles will be present here. Here absent. Chloroplast present here. Here absent. Okay. So these are all the differentiation of prokaryotes and eukaryotes. So if you are thorough in this differentiation, you can able to answer many questions regarding prokaryotes and eukaryotes. Can you all understand, children? Okay. If you have any doubts, please ask me. So let us now look at individual cell organelles to understand their structure and function. So after ongoing subject, we are going to study about an individual cell organization, cell structures and their functions. Okay. So next we are moving on to the cell membrane. So what is cell membrane? What is cell membrane? What is cell membrane? Tell me, anyone can tell. Cell membrane. We are going to see now as cell membrane. So the detailed structure of the membrane was studied only after the advent of the electron microscope. So in 1838 Matthias was discovered, 39 Theodore was discovered. So after, but they were all discovered cell, what is cell, and who gave the final shape? Richard which was given a final shape, because after 1900 only he was because 1855 sorry, and after that also no one can able to see the cell. So after coming up of electron microscope, that is 1900 only electron microscope was invented. So after that only cell membrane was fully can be able to see in the electron microscope, and it gives the detailed structure while we seeing in the electron microscope. So electron microscope in the 1950s. So meanwhile, chemical studies on the cell membrane, especially in human red blood cells, that is RBC, erythrocytes, enable the scientists to deduce the possible structure of plasma membrane. So in 1950s, they were studied in detail about cell membrane in the electron microscope. After that, meanwhile, chemical studies also experiments were done by the chemical studies through human red blood cells. Human red blood cells, the question may ask by human red blood cells, RBC or erythrocytes. So three are same. So in this human red blood cells, the scientists can deduce, that is, what is deduce? A conclusion. Conclusion is called reduce. So the scientist was concluded that possible structure, that cell membrane is having enough structure to study. So these are all the conclusion who have told by the scientist. Okay. So then these studies showed that the cell membrane is mainly composed of lipids and proteins. This membrane, this full membrane, cell membrane was fully covered by what? Lipids and proteins. Lipids and proteins. So here proteins and lipids were present in the cell membrane. Lipids and proteins are present in the cell membrane. CM, I am just putting like this. And then next, the major lipid are phospholipids that are arranged in a bilayer. So all lipids are phospholipids that will be arranged in the bilayer. We are seeing in the we will see bilayer. What is phospholipid bilayer? So in this phospholipid bilayer, we will having polar head and non-polar head. So phospholipid bilayer. Here it is having polar and non-polar head. What is polar and non-polar? How it is like? So we will see. Okay. Polar and non-polar head. Also the lipids are arranged within a membrane with the polar head. We have seen, no? This is lipid was present in the phospholipid. So it is having an bilayer. Bilayer means two layer it is having. So one is polar head. That one is polar head. Polar head is water acceptance. Polar head will accept the water resistance part towards the outer side of the hydrophobic tails. Hydrophobic tails is non-polar head. Hydrophobic tail is non-polar head. This is also called as hydrophobic. Hydrophobic means what? Hydrophobic. Hydro means water, phobic means panic. So, this is in the polar அப்படின போது அது வந்து water resistant. Water வந்து accept பண்ணிக்குது. So, whereas non-polar head na அப்ப இங்க என்ன ஆகாது water? Accept பண்ணிக்காது. So, it is having polar head and non-polar head like this. This is bilayer. This is bilayer. இப்போ இந்த ரெண்டு bilayerம் எப்படி இருக்குன்னு நம்ம பார்க்குறோம் அப்படின்னா it is parallel இருக்காங்க. ஓகேவா? இப்படி இருப்பாங்க. So, இந்த மாதிரி parallel இருக்காங்க. So, இப்படி இருக்கும்போது this is an polar head and this is an hydrophobic tail. Hydrophobic tail is non-polar head. So, polar head and non-polar head towards the inner part. So, this is the hydrophobic tail is an inner part, whereas polar head is an outer part. This ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. You can see in the picture figure 8.4. So, it is a fluid mosaic model. So, and scientist was proposed a fluid mosaic model. So, in this model you can see the upper part. So, here it is. So, this is an lipid and here one lipid will be present. So, இப்படி நம்ம பார்க்கும்போது இங்க வந்து peripheral lipid இருக்கு and then integral. This is an integral middle layer. So, once this is a cell membrane. If in the cell membrane la நம்ம lipids நம்ம பார்க்குறோம் அப்படின்னா phospholipids we can able to cut it off, but in the integral lipids we can't able to. If we will cut the integral lipids அப்படின்னா the whole cell membrane will be collapse இல்லையா? ஸோ அது ஃபுல்லாகவே டேமேஜ் ஆனால் தான் இன்டீக்ரல் ப்ரோட்டீனை நம்மளால் வெளியில் எடுக்க முடியும். ஸோ வேரஸ் இந்த பாஸ்போலிபிட் நம்ம பார்க்குறோம், பாஸ்போலிபிட்னா பெரிஃபரல் லிபிட் நம்ம பார்க்கும்போது we can cut it off, right? ஸோ இதை தான் நம்ம சொல்கிறாங்க. And then in addition to phospholipid membrane also contains cholesterol. This phospholipid also contains what? Cholesterol. Then next, later biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrates. So, first chemical studies will be done by the scientist. Later on biochemical investigation will be done. So, biochemical, biology as well as the chemistry was investigation done. After that investigation, the scientist was told that cell membrane also contains what? Not only protein and fat, not only lipids and protein, it also contains what? Carbohydrates. Carbohydrates also this cell membrane containing. So, after that the ratio of protein and lipid varies considerably in different cell types. So, cell types, different cell types is different cell types is containing different proteins, carbohydrates, lipids and all, because in 100% we can able to see. The ratio of protein and lipid varies considerably. In human beings, the membrane of the erythrocyte is approximately 52%. So, in human beings we can see in human beings, RBC, that is erythrocytes, in human beings, erythrocytes is approximately 52% of proteins. Human beings we are seeing. So, protein present 52%. Then, after 52% protein and 40% lipid. Lipid present in 40%. So, altogether, so we will see lipid present in 40%. Depending on the seas of extraction, membrane protein can be classified as integral and peripheral. Peripheral proteins lies on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. We have seen. So, this is an cell membrane, this is an integral protein, so this is an phospholipid, peripheral lipid. So this is on surface, this is in the inner part of the cell membrane. So it will be, if we can take this lipid and all, we have to collapse, we have to calibrate, we have to bury the cell membrane. After that only we can able to pull it off the lipid from here. But peripheral lipid we can able to cut it off from the surface. So here in human beings, red blood erythrocyte is having all cell membrane is having protein, lipid and carbohydrate, and in your NCERT it is given only protein and lipid. So protein is present in 52% and lipid is about 40%. So altogether 92%. So rest of 8% what? Rest of 8%, that is an carbohydrate. So carbohydrate, because carbohydrate also present in the cell membrane, right? So 8% will be present in the carbohydrate. So this is cell membrane. And then next, an improved model of the structure of cell membrane was proposed by Singer and Nicholson in 1972. So in 1972, Singer and Nicholson is a scientist who discovered fluid mosaic model. Fluid mosaic theory was proposed by Singer and Nicholson in 1972. This will be asking for your NEET exam. And widely accepted the fluid mosaic model. So we have already seen the fluid mosaic model, that is this structure, cell membrane, integral protein, peripheral protein, we have seen already. According to this, the quasi-fluid nature of lipid enables later movements of proteins within the overall bilayer. This ability to move within the within the membrane is measured as a fluidity. So what is quasi-fluid in this fluid mosaic? Singer and Nicholson was proposed the fluid mosaic theory. So அப்ப அந்த fluid mosaic theoryல என்ன இருக்கு அப்படினு பார்க்கணும்னா quasi-fluid. What is quasi-fluid? Quasi-fluid means what? A jelly-like substance. A jelly-like substance, that is quasi-fluid. அப்போ இந்த jelly-like substance இருக்கிறதுனால ஈஸியா, because it is an motile இல்லையா, so flagella அது மூவ் பண்ணணும். ஸோ அப்போ அங்கே மொட்டைலாக இருக்கிறதுனால அங்கே quasi-fluid இருக்குது. ஸோ this quasi-fluid is helping for the movements. அப்போ மூமெண்ட்ஸாக இருக்கும் போது அப்போ அங்கே fluid, இதுவே ட்ரையாக இருக்குது அப்படின்னா அங்கே மூமெண்ட் இருக்குமா? இருக்காது ரொம்ப. இப்போ ஃபார் எக்ஸாம்பிள் நான் சொல்கிறேன், இப்போது யூ ஆர் ஆல் ஈட்டிங் சம் ஃபுட். ஸோ அப்போ ஃபுட் எடுக்கும் போது நம்ம என்ன பண்ணுறோம், சாப்பிட்டுட்டே இருக்கோம். அந்த இடத்துல நம்மளுக்கு என்ன ஆகுது, இப்போ இதுவே இங்கே நம்மளோட ஈசோஃபேகஸில் என்ன இருக்கும், எதுவுமே ட்ரையாக இருக்குது அப்படின்னா என்ன ஆகும், நம்மளுக்கு என்ன ஆகுது, உள்ளே போகுமா, ரொம்ப அடைச்சிக்கும், ஒரு மாதிரி இருக்கும். ஸோ அந்த இடத்துல யூ கேன் வாட், யூ கேன் ஹாவ் வாட், சம் ட்ரிங்க், இல்லை தண்ணி குடிக்கணும். அப்போ தண்ணி குடித்தா தான் ஈஸியாக உள்ளே போகணும், பிகாஸ் அங்கே என்ன இருக்குது, அப்போ ஃப்ளூயிட் இருக்கணும். ஸோ திஸ் இஸ் அன் எக்ஸாம்பிள். ஸோ அதே மாதிரி தான் செல் வால் கண்டெய்ன்ஸ் திஸ் குவாசி ஃப்ளூயிட். அப்போ அந்த ஃப்ளூயிட் இருந்தால் தான் அங்கே என்ன ஆகும், மூமெண்ட்ஸ் அழகாக கரெக்டாக இருக்கும். ஸோ அதனால் மூமெண்ட்ஸ் இருக்குது அப்படின்னு சொல்கிறாங்க. ஸோ அப்போ அந்த மெம்ப்ரெயினில் ஃபுல்லாக என்ன இருக்கும், ஒரு ஃப்ளூயிடிட்டி இருந்துகிட்டே இருக்கும், ஒரு அதாவது ஒரு அந்த ஃப்ளூயிட் ஜெல்லி லைக் சப்ஸ்டன்ஸ் இருந்துகிட்டே இருக்கிற மாதிரி தான் இருக்கும். அண்ட் தென் நெக்ஸ்ட் பாயிண்ட், The fluid nature of the membrane is also important from the point of view of function like cell growth. This fluid in the quasi fluid எதுக்கு helpful இருக்கும்? Cell growth ஆகிறதுக்கு. So it helpful for cell growth, formation of internal cellular junctions and secretions of some enzymes, and then endocytosis. What is endocytosis? Endo means what? Endo means inside, cytosis means cytoplasm. So inside the cytoplasm it is helping, and then for cell division also this fluidity is helping. So fluidity is helping. Fluidity have the function of cell growth, formation of intercellular junction. Some junctions will be there, no? That is an intercellular junction, and secretions, endocytosis, and for cell division it is helpful, it is formation. And then next, one of the most important function of the plasma membrane is the transport of the molecule across it. So one most important function of cell membrane is what? It is a transport material. It is an what? The cell membrane is a transport material. So the membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called a passive transport. So here we are seeing passive and active transport. So in passive transport it is having some energy, it can take some energy. So energy will be produced in the form of what? ATP, adenosine triphosphate, that is ATP. Okay. So in the form of ATP while passive transport, and another one is in the cell membrane active transport. So in passive transport energy will be taken, but in active transport energy doesn't takes place. What? Yes, sorry. So and then in passive means energy what is without any requirement of energy, passive will be takes place. So that is an diffusion, osmosis, that is higher concentration to the lower concentration, that is an passive energy. So passive transport, passive energy and active energy. So in passive without energy, in active with energy, energy wants to active transport, and for passive we can't able to give energy because energy வந்து யூஸே ஆகாது passiveல. So passive and active அப்படின்னும் போது அப்போ அங்கே osmosis இருக்கு. This is higher concentration to the lower concentration, high to low. Active passive அப்படின்னு நம்ம எடுத்துக்கிட்டோம் அப்படின்னாலே this is high to low, whereas high to low, lower concentration, whereas active energy நம்ம பார்க்குறோம் அப்படின்னா low to high concentration. அப்போ this low to high in the sense, for example நான் சொல்கிறேன் என்ன அப்படின்னா, இப்போ we are stepping down from the upstairs. ஸோ இப்போ நம்ம ஒரு படிக்கட்டிலேருந்து கீழே இறங்குறோம் அப்படின்னா we can நம்மளுக்கு எனர்ஜி லாஸ் ஆகுமா? இப்போ ஒரு ஸ்டெப்ஸ் இருக்குது, இப்போ நம்ம ஸ்டெப்ஸ்லேருந்து கீழே இறங்கிட்டே இருக்கோம், இறங்கும் போது எனர்ஜி லாஸ் ஆகுமா? எனர்ஜி லாஸ் ஆகாது. இதுவே நம்ம ஸ்டெப்ஸ் ஏறி போகிறோம், மேலே அப்ஸ்டேர்ஸ் ஆகிறோம். ஸோ அப்ஸ்டேர்ஸ் ஏறும் போது நம்மளுக்கு என்ன ஆகுது? ஒரே நம்ம பால்பிட்டேட் ஆகும், ப்ரீதிங் டிஃபிகல்ட்டி இருக்கும். ஸோ அப்போ எனர்ஜி நம்மளுக்கு தேவை, அங்கே நிறைய நம்மளுக்கு ஸ்வெட் ஆகும் இல்லையா? ஸோ அப்போ இது லோயர் கான்சன்ட்ரேஷன் டு ஹையர் கான்சன்ட்ரேஷன். அப்போ லோயர் கான்சன்ட்ரேஷன்லேருந்து ஹையர் கான்சன்ட்ரேஷன் வேணும்னா அங்கே என்ன தேவைப்படுது? எனர்ஜி வந்து யூட்டிலைஸ் பண்ணிக்கிறோம் இந்த ஃபார்ம் ஆஃப் எப்படி? In the form of ATP, that is an active transport. அப்போ passive transportனா higher concentration to the lower concentration, that is osmosis. அப்போ அந்த இடத்துல நம்மளுக்கு என்ன தேவையில்லை? எனர்ஜி தேவையில்லை. அப்போ அங்கே வித்தவுட் எனர்ஜி நம்மளுக்கு ஹையர் கான்சன்ட்ரேஷன்லேருந்து லோயர் கான்சன்ட்ரேஷனுக்கு எனர்ஜியே தேவையில்லை, தட் இஸ் passive transport. This is active and passive. Okay, you people able to understand? This is cell membrane. Cell membrane இதோட நம்மளுக்கு முடிஞ்சுது. So and then we'll go for cell wall. So cell wall. So after cell wall we can finish the class. Okay. So cell wall, as you may recall, non-living rigid structure called the cell wall forms an outer covering of the plasma membrane of fungi and plants. So cell wall, so nearly it is too late, I think so, because 9:30 your class, next class is going on, right? So tomorrow we can see the cell wall, okay, cell wall and endomembrane system. So we can finish endomembrane system till tomorrow. So let's start the class from cell wall from tomorrow, students. So if you have any doubts, please please ask me tomorrow and go through this session. So tomorrow we'll have some recap and then we'll move on to the cell wall. Okay, thank you, students.